Welcome back.
With the general microscopic structural background, which was discussed in the previous slides, depending upon the type of acini, amount of connective tissue, and number of excretory ducts, the glands are classified into three types: serous salivary gland, mucous salivary gland, and mixed salivary gland. Do not get confused with the classification of epithelial glands. Both are different. I will repeat, epithelial glands classification is based on arrangement of secretory cells and duct system. But salivary glands classification is based on the presence of predominant type of acini, amount of connective tissue, and number of excretory ducts. We will now see the details of three types of salivary glands. As already discussed in the previous slide, the three major salivary glands are parotid, submandibular, and sublingual. These three glands are located outside the oral cavity and convey the secretions into the mouth through large excretory ducts. The histology of the major salivary glands are best examples of compound tubulo-acinar glands. As the name suggests, the secretory elements of these glands are shaped in the form of acini and tubes. Parotid consists of serous acini. The submandibular and sublingual salivary glands contain both serous and mucous acini, but predominating with one type of acini. Ducts are distinct structures in the salivary glands. They are lined with cuboidal, columnar, or stratified epithelium and are named according to their location in the gland. Now let us discuss in detail all the three major salivary glands. Example for serous salivary gland is parotid gland present in the cheek antero-inferior to the ear. The parotid salivary gland is a large serous gland classified as a compound tubulo-acinar gland. That is, it has branched duct system with secretory cells arranged in the form of tubular and acinar pattern. In this diagrammatic illustration, a section of the parotid gland is depicted at lower magnification. The parotid gland is surrounded by a connective tissue capsule from which arise numerous septa that subdivide the gland into several lobules. Located in the connective tissue septa between the lobules are small arterioles, venules, interlobular excretory ducts, and numerous adipose cells. The amount of intralobular connective tissue between the acini is very little. Each lobule consists of cross sections of closely packed masses of secretory cells, the serous acini, the intralobular ducts, and sometimes adipose tissue. As discussed earlier, the serous acinus consists of pyramid-shaped cells arranged around a small lumen. The spherical nuclei of these cells are located at the base of the slightly basophilic cytoplasm. Small secretory granules are visible in the cell apices called zymogen granules. The number of secretory granules in these cells varies with the functional activity of the gland. The serous acini are also surrounded by thin, contractile myoepithelial cells located between the basement membrane and the serous cells. Usually, only their nuclei are visible. The secretory acini empty their product into narrow channels, the intercalated ducts, which are longer. These ducts have small lumina lined by simple squamous or low cuboidal epithelium and are often surrounded by myoepithelial cells. The secretory product from the intercalated ducts drains into larger striated ducts which are longer in length. These ducts have larger lumina and are lined by simple columnar cells that exhibit basal striations. To recall, the striations are formed by deep infolding of the basal cell membrane. The striated ducts empty their product into interlobular excretory ducts that are located in the connective tissue septa. Their lumina become progressively wider and lined by tall columnar epithelium. From the interlobular duct, the secretions enter into the single large main duct, which is lined by stratified columnar epithelium, and near the opening into the oral cavity to pour the secretions, it is lined by stratified squamous non-keratinized epithelium. Serous cells of the parotid gland secrete abundant alpha amylase that initiates into the hydrolysis of carbohydrates and proline-rich proteins, which has antimicrobial and protective properties. We will now see what are the specific features of the serous salivary gland or parotid gland apart from the general features like capsule, connective tissue septa, dividing the gland into lobes and lobules, and the excretory duct system, which includes intralobular and interlobular ducts. In specific features of the histological section of the gland, we are looking into type of acini, whether serous or mucus, amount of connective tissue if it is abundant or less, and number of ducts, whether it is more or less in number. If the duct is longer, more number of ducts can be observed. If the duct is shorter, less number of cross sections of the ducts will be observed, or the ducts may not be seen in the section. In the parotid or serous salivary gland, the specific features are it has predominantly serous acini, which are closely packed. The intralobular connective tissue between the acini, the interlobular connective tissue between the lobules is very minimal or less, and also it has less adipose tissue. Intralobular ducts, which includes intercalated ducts and striated ducts, and interlobular ducts are more in number because of the longer length. Now let us look into the second type of gland, the mucous salivary gland. Example for mucous salivary gland is sublingual gland, present in the floor of the oral cavity. This is the section under low power. The sublingual gland is a compound mixed tubulo-acinar gland. The gland is covered by connective tissue sheath called capsule. The interlobular connective tissue septa arise from the capsule and divides the gland into lobules. It is more abundant in the sublingual than in the parotid and submandibular glands. Numerous arterioles, venules, nerve fibers, and interlobular excretory ducts are seen in the septa. This gland resembles the submandibular gland. Each lobule is composed of both serous and mucous acini, vessels, and intralobular ducts. The light-stained mucous acini are more prominent with very few serous acini. In the section, the serous acini may be seen or may not be seen since the number is very low. The composition of the gland is variable. The myoepithelial cells are seen around individual acini. In comparison to other salivary glands, the duct system is somewhat different. Typical intercalated ducts are infrequent or are absent in the sublingual gland since the length of the intercalated ducts are short or absent. When present, it is lined by simple squamous or low cuboidal epithelium and are not readily observed in a given section. This is because the section might not pass through the intercalated ducts since they are very short. These intercalated ducts join to form non-striated intralobular excretory ducts, which are more prevalent in the sublingual glands. These ducts are equivalent to the striated ducts of the submandibular and parotid glands, lined by simple columnar epithelium but lack the extensive basal striations. Okay, so because they lack the basal striations, they are called as non-striated ducts. These ducts join to form the interlobular excretory duct lined by tall columnar epithelium. The interlobular ducts join to form single large main duct which is lined by simple columnar epithelium to stratified epithelium and open into the oral cavity to pour the secretions. At the terminal end where it opens into oral cavity, it is lined by stratified squamous non-keratinized epithelium. The specific features of mucous salivary gland or sublingual gland are both serous and mucous acini are present, but mucous acini predominates over very few serous acini. The connective tissue between the acini and interlobular connective tissue septa are abundant. The intercalated ducts are short or absent and not observed in the section. The non-striated intralobular excretory ducts. The non-striated intralobular excretory ducts are numerous and equivalent to the striated ducts of submandibular and parotid glands, but lack extensive membrane infolding and basal striations. And also, the interlobular excretory ducts are prevalent or more in number. The few serous cells present in demilunes add amylase and lysozyme to the secretion. The mucous cells secrete the viscous mucus which help in lubrication. Now coming to the third type of salivary gland, that is mixed salivary gland. The example is submandibular gland which is present deep to the body of mandible. In this illustration, a section of the submandibular gland is depicted at lower magnification. Like the parotid salivary gland, the submandibular is also a compound tubular and acinar gland. So compound tubulo-acinar gland. Like in the parotid gland, the connective tissue surround the gland and form the capsule. Thin interlobular septa arise from capsule and divide the gland into lobules. The amount of connective tissue is moderate compared to parotid gland. Numerous interlobular excretory ducts of different sizes are located in the interlobular connective tissue septa. Also located in the connective tissue septa are nerves and numerous different sized arterioles and venules. Numerous adipose cells are seen scattered among the various acini and in the connective tissue septa. Each lobule of the submandibular gland has few mucous acini intermixed with numerous serous acini. We have discussed lots about this. Just to recap, the serous acini are similar to those observed in the parotid gland. These acini are characterized by smaller size, darker stained pyramidal cells, spherical nucleus located basally, and secretory granules in the cell apices visible at higher magnification. The mucous acini are larger than the serous acini and are more variable in their size and shape. The mucous cells are more columnar and exhibit pale or almost colorless cytoplasm after routine histologic staining. The nuclei are flattened and pressed against the base of the cell membrane. The mucous acini also have a somewhat larger and more apparent lumina. The third type of acini is mixed acinus called serous demilune or the demilune of Giannuzzi. They are normally mucous acini surrounded or capped by one or more groups of serous cells forming a crescent-shaped serous demilune. Recently, it is suggested that the aggregates formed at least in part is an artifact formed when the adjacent mucous cells swell during routine tissue preparation. The thin contractile myoepithelial cells surround the serous mucus and intercalated duct cells. The duct system of the submandibular gland is similar to that of the parotid gland. The intralobular intercalated ducts are few in number and have small lumina and are shorter lined by simple squamous or low cuboidal epithelium, while the striated ducts are lined by simple columnar epithelium or longer than in the parotid gland and are observed more in number in the sections. The secretions from the striated ducts drain into interlobular excretory duct lined by tall columnar epithelium, and these ducts join to form the single large main excretory duct, which is lined by striated columnar epithelium and open into the oral cavity to pour the secretions. At the terminal end where it opens into oral cavity, it is lined by the stratified squamous non-keratinized epithelium. The submandibular salivary gland produces 2/3 of all the saliva, which is poured into the oral cavity. The secretion of each gland is either serous, seromucus, or mucus, depending on its content of the glycoprotein mucin. Saliva from the parotid is serous and watery. The submandibular and sublingual glands produce a seromucus secretion, while that of the minor glands is mostly mucus secretion. The specific features of mixed salivary gland or submandibular salivary gland are it has serous acini, mucus acini, and serous demilunes. The serous acini predominate in the glandular tissue. Intralobular connective tissue between the acini is moderate, but interlobular connective tissue septa is well developed. It has few intercalated ducts and more of striated ducts. We are now in the last section of epithelial glands and salivary glands, that is clinical anatomy. Cyalorrhea, the excessive saliva production, is associated with the autonomic activity of nausea, inflammation within the oral cavity, and rabies viral infection. The second clinical condition which can be observed in salivary gland is the xerostomia, which is inadequate salivary production leading to dry mouth, can be caused by various factors affecting the major salivary glands such as mumps, viral infection, radiation of glands, or the normal side effect of drugs such as antihistamines. The third type of the clinical anatomy of the salivary glands is tumors of salivary glands. Pleomorphic adenoma is one type of salivary gland tumor, which is the mixed salivary tumor, is a common tumor of major and minor salivary glands. It is characterized by pleomorphic or mixed appearance in which there are epithelial elements forming the inner layer of cysts and tubules, myoepithelial cells as the outer layer of cysts and tubules, both scattered within the stromal component, which is typically of mucoid, myxoid, and chondroid tissue. It can also be hyalinized or fibrotic. The other type of tumor of salivary gland is mucoepidermoid carcinoma, which is the most common malignant salivary gland tumor. The tumor is composed of combination of 4 types of cells, that is mucin producing, squamous, intermediate, and clear cells. Well differentiated tumors have predominance of mucus cells, while poorly differentiated have more solid and infiltrative pattern. So with this we come to the end of the salivary glands. To recap, we have discussed about the general structure of the salivary gland, in part 1, and in this second part we discussed about the classification of the salivary glands and the details of each salivary gland, concluding with the clinical anatomy of the salivary glands. Okay, so in the next section or the session we will discuss, okay, I'll be discussing about the summary of the whole epithelial glands and the salivary glands. Okay, see you there. Join my Facebook community, link in the description. Like, share and subscribe. Thank you for watching.